Here doing a video on AS biology on cell division and mitosis. So let's get straight into it. So this just comes under the cell theory, and that is the idea that all new cells arise from pre-existing ones due to mitosis. Every cell in your body would have started as one cell called a zygote when fertilization first occurred, and then basically you obviously keep dividing, you keep making new cells. Mitosis is actually only nuclear division, so it's only what happens in the nucleus. So before we look at mitosis, we're going to look at the whole of the cell cycle, and it is, don't forget, mitosis is about creating an identical set of cells. So it's not making the gametes, not making the sperm, egg, or pollen cells. And mitosis, the point of it is for growth, replacement of cells, and asexual reproduction of things like bacteria. So chromosomes, then, you need to know what a chromosome is. So chromosomes obviously replicate as DNA replicates. The DNA chromosome is just a long strand of DNA that is wrapped around a histone protein. So you need to know that word, you need to understand that's that. And that's why when we talk about bacteria cells, prokaryotic cells, we say it's naked DNA, which isn't wrapped around a histone protein. Now, what happens basically is we start off with two individuals. So these are two different chromosomes, chromosome 1, chromosome 2. It might be in a pair, so they might be the one that you got from your dad, let's say. That might be the one that you got from your mom. Now, we want to replicate both of those, so there'd be 46 of those 23 pairs in our body, and they get replicated to make these, and we create something called sister chromatids in here. And you might be able to hear my daughter eating an apple next to me. So we're going to be looking at the cell cycle in eukaryotic cells, which are multicellular organisms. It's very different in prokaryotic and bacteria and viral cells because they are different to us. But obviously we're looking at the whole cell cycle. So mitosis, as I said, is just the nuclear division stage. So we've got to have this whole stage of interphase happening in order to prepare the cells to divide. So when the nucleus divides, everything is ready to start the new cell. So we have a look at what happens through the stages. The first main part is G1 stage, which is where the cells grow bigger and they get new organelles and proteins needed to replicate the DNA and to become two cells. At the S phase, we replicate the DNA, and at the G2 phase, that is when some new proteins are made, but also the cell just grows as well. So different versions of cell cycles that you might see, you might have to calculate times using pie charts and things like that. But just remember, interphase is not technically part of mitosis. It is part of the preparation in the cell cycle. And there is also an extra phase called G0 phase, which is where cells go into a rest phase between doing the cell division and the cell cycle. So we need to learn then what mitosis is, what is happening at each stage. So if we have a look at this, we've got interphase technically isn't part of mitosis, as I said before, but I always include it in when I think about it because I remember the word if-mat, and I sort of talk my way through it. So just to start off then, what's sort of happening here, we've got mitosis, as I said, technically not really starting until probably just before there, but never mind. So interphase, we said the DNA replicates the DNA, the organelles are made outside the nucleus, okay? And at the end of interphase phase, start of prophase, the chromosomes, the lengths of DNA supercoil, which means they shorten and get thicker, and that means that the DNA is able to be moved around, and as a result can be manipulated without getting damaged. We then enter the stage called prophase. So prophase is when the nuclear envelope starts to deteriorate, the chromosomes, as I said, are supercoiled, okay, and as a result, the other thing that happens is the centrioles move to the poles. Then in metaphase, what happens is the chromosomes line up along the equator, and in the centromere, which is the center of the chromosome, the spindles attach to the centromere in the center of the chromosomes. Then what will happen is the spindles contract, pulling the chromosomes to each opposite end of the pole, and in anaphase, that is where your chromatids, sister chromatids, which are genetically identical to each other, separate. So this one goes that way in a V shape, that one goes that way, and it's being pulled by the spindle fibers. Okay, and they go back to their opposite poles. In telophase, which may or may not include cytokinesis, is when the nucleus reforms, because the nuclear envelope reforms. The chromosomes will uncondense, unsupercoil, and go back to the cells. And as a result, you form two nucleuses. And obviously, that can then continue, and the cell can split into two cells, which is called cytokinesis. And that's the overall stages of mitosis. So let's have a look at them in a bit more detail, and this is sort of facts that you'd need to be expected to know. So interphase, cell signaling, the cell signal reaches the cell, tells the DNA to replicate. Chromosomes make a copy of themselves, and the DNA is replicated and is then checked for mutations. Prophase, they start to coil up, they become thicker and shorter and can now be seen with an optical microscope, which is why we then start saying this is mitosis. Nuclear envelope breaks down and centrioles move to the opposite ends of the poles. Metaphase, the chromosomes line up along the equator, spindles attach to chromosomes at the centromere, and they pull apart. And you can see here on the diagram, the chromosomes normally stain a darker colour, so you can see here the dark colour, the staining of the chromosomes. So you can see that in the required practicals that you have to do, where you see metaphase actually happen. Then you've got anaphase, which is where the centromeres pull apart and the sister chromatids move to the opposite ends of the poles. And again, you can see here this quite clearly on a micrograph here. You've got one set of chromosomes being pulled that way in the V shape and one being pulled that way in the V shape as the spindles contract. The chromosomes then reach the pole in telophase and the nuclear envelope reforms. You can see here a really clear diagram of that in on a micrograph here for you as well. We then get to cytokinesis then. So this is obviously doesn't necessarily always happen immediately. So it's sort of like is part of mitosis, but not always included. But you do need to talk about it as part of at the end of telophase. And basically, the cell divide into two. And animal cells, this happens because the cell membrane is pulled in and it pinches off. So you get something called the cleavage furrow forming there, which is when basically the membrane of the cell pinches inwards that side, pinches inwards that side, and then it splits into two separate animal cells. Now that then is different from plant cells because the plant cells form a different way. So what happens is in plant cells, vesicles fuse across the cell, and then they get layers of cellulose laid down across them, making a new cell wall, and we call this a cell plate is formed, splitting the two cells into two. So there's no cleavage furrow there. Okay, so a little bit of maths then that you might be asked to do is work out the mitotic index. Now, I've linked this to percentage change calculations because percentage change, whether it's increase or decrease, is always the actual change. So if I said a population has dropped, population of 200 rabbits has dropped by 50, you'd do the actual change, 50, divided by the original amount, so it'd be 50 divided by 200, okay, times 100%. Mitotic index is similar as well. Mitotic index is basically how many cells of the total cells are doing the process of mitosis. So don't forget the cell cycle interphase isn't mitosis. So if you look at this pie chart, you could get something like this where 55% of the cells are doing interphase, so that means 45% of the cells are doing mitosis. Or you could get a maths question like this one at the bottom. So if we look at this, mitotic index is the number of cells doing mitosis divided by the total number of cells counted. So if you have a look at this question, our total number of cells counted is 200. So it says in a sample of 200 cells, 8 are in prophase, 14 are in metaphase, 5 are in anaphase, and 9 are in telophase. What is the what is a mitotic index, and then how do you calculate mitotic index? So the mitotic index is the amount of cells or the percentage of cells that are completing mitosis in the sample counted. So here, if I add up 8, 14, 5, and 9, that would give me a total of 36, and there's 200 being counted. So therefore, my mitotic index is 36 divided by 200, which equals 27% of cells, or 0.27, because it's not actually after percentage. And here we got, just to finish off then, some cells doing cytokinesis. So you can see you could get lots of different versions of the cartoons that you might need to use to answer the questions. So let's see if we can recognize the stages. So first of all, we've got this one, which hopefully you know is anaphase. Then we've got this one, which would be in interphase, because you can't see anything particular happening. This one, which would be in metaphase, because the chromosomes are lined up along the equator. This one, which would be, quite a tricky one there, I'd say anaphase, in prophase, because the prophase, the chromosomes are coiled, but it's one nucleus and not a lot else happening. Then you've got one which we'd either describe as telophase or cytokinesis. Okay, and again, probably cytokinesis, that one. And that one, really hard to see, that one, not clear enough for an example. I'd say probably prophase, maybe late interphase, early prophase. And again, you've got metaphase there, anaphase, where the chromosomes being pulled to their poles, and then anaphase again on that one. And that one looks like metaphase because the chromosomes are in the center of the